function. I'm assuming there's a relationship there, but it is a pretty big assumption. I realize that. Okay, so when I think of sunflowers, I think of this. You might think of something different. So this is the common sunflower. This is Helianthus annuus, the one that we domesticated. And historically, we think that the range of this species was restricted to the Great Plains of North America. Probably up into Canada, but definitely not very far east, not in North Carolina, and not very far west, probably not over the Sierras. But sometime during the last couple thousand years, we think that humans likely introduced this species into California and also throughout North America. And so, today, we observe a new species in California. This species has now been named, it's Helianthus winteri, and unlike annuus, the ancestor, which is annual and relatively herbaceous, this species is woody and it's a perennial. And it also has aseasonal flowering, so it starts flowering and it basically doesn't stop flowering during its lifetime. And it puts on a lot of really dense wood. The species has a relatively restricted range currently. It's found just on the western slopes of the Sierra Nevadas, the sort of Sierra Nevada hillsides between about 50 and about 300 meters of elevation. It's only found on ungrazed, northern-facing, granite-decomposed slopes. We don't know the historical distribution. But you can see here the current distribution of annuus is more or less parapatric with that. You don't see them growing in the same place, but you have populations within about 100, within about 1 kilometer of each other. Okay. So winteri is different in a number of really interesting ways, one of which is the woody tissue that it produces. And you can see that here. You can see it very early on. So these are stem sections from 3-week-old seedlings. On the right there for you guys is annuus, a typical annuus stem, and it's very sort of well-ordered development of vascular bundles. And it's just starting to put in some secondary growth here between its vascular bundles. Winteri, on the other hand, grows a bit more slowly, but it starts developing secondary growth a lot more rapidly. It's a bit more disorganized in the way that it develops it. And this continues through the lives of the plants. So these are stem sections taken from adult plants. These two were grown in the same common garden. Here is annuus, here is winteri. And you can see that at the base of the stem, they're both relatively woody, although the wood here is denser. But as you go up the stem, about 1 meter up the stem, annuus becomes mainly pith, winteri remains pretty woody. And when you take sections from plants in the wild, you can see that they're very woody, and they're perennial, so they continue to grow and put on more wood year after year, and you can actually discern growth rings in this plant. Okay, so what we did is sort of a preliminary analysis to begin to describe variation and divergence in the system is take 6 seedlings of each species and sequence transcriptome from all the above ground tissue at about 3 weeks. So it's just pretty simple collecting of one life stage grown in a common environment. So we took that transcriptome sequence and we aligned it to the annuus transcriptome. We identified SNPs, and we calculated FST per SNP as an estimate of the coding sequence divergence between the 2 species. And we also identified significantly differentially expressed genes using, or I guess, contigs using DE-seq in our package. Okay, so this is just descriptive. You've seen plots like these many times before, I'm sure. But this is just showing that they're, here in red are the dots that represent genes that are differentially expressed on this log scale for fold change between the 2 species. These things are more expressed in winteri, these things are more expressed in annuus. And overall, we see about 3.5% of the genes that we have are differentially expressed in seedlings. Okay, so that's pretty descriptive, not all that interesting necessarily. We thought maybe we'd try to figure out a little bit more about what these genes were, what they were doing. So we performed a Go analysis. And using this program RMINJ, which was developed at UBC, basically, as opposed to sort of a traditional Go analysis where you draw a cutoff and you compare things above the line to below the line, this particular approach that we use, the receiver operator characteristic approach, just sort of counts your ranking. It's a ranking-based score. So here you can see that the blue lines are ranked higher on the list than they are here. So when we do that, We get 7 categories, biological process ontology terms, that are overrepresented in our differentially expressed genes. Some of them we can't really tell a story about, and all we can really do, of course, is tell a story. So nucleosome organization, maybe DNA packaging is important, I don't know. There's a few, though, that we can kind of tell a story about. So response to gibberellin stimulus, gibberellin, broadly involved in many aspects of plant development, including stem lignification and lignobiosynthesis, so maybe a woody story there. And phenylpropanoids also sit upstream from many biosynthetic pathways, including the production of flavonoids and lignans. So, okay, again, story could be sort of cool and interesting. But we were interested in comparing those data to divergence and coding sequence. And so that we could have comparable data sets, we just did a cutoff. We took the top 3.5% of genes, the percentage that had been differentially expressed, with either the highest mean FST, so averaging all SNPs across a gene, or single SNPs within genes, the highest maximum FST. So we have those 2 different measures of FST, and they're slightly different lists. Among those, there are 2 genes that are quite promising for candidates for explaining the divergence between these 2 species. One is a homologue of Arabidopsis flowering BHLH. It's a transcription factor involved in regulating flowering time. And we know that many of the regulators of the flowering time network are also involved in regulating transcription during secondary growth. So could be something interesting, an avenue to explore. We also see a homologue of this xylan biosynthesis gene, and xylan is a component of cell walls, and again, sort of a lignin story, a woody story is emerging here. And we performed the same ranking ontology test with these data. Didn't get anything significant. I think it's because FST or null or neutral distribution is different than the expectation under this particular test. But just want to point out something particularly interesting to me is that we're seeing coding changes, divergence in coding of genes that seem to be involved in regulating things in which we see divergence in expression. So that may be suggestive that maybe we're seeing sequence changes here that may cascade down to expression changes here. But again, very descriptive. For me, the key story is this. When we compare the genes that are diverging in coding sequence to the genes that are diverging in expression in these 2 species, we see very few genes, in fact, no more than we would expect by chance here in the overlap. And to me, that suggests that, at least in this particular system, any particular gene is either evolving via one path or the other. That compares somewhat, contrasts somewhat with data we have from more divergent taxa. So there's data from Drosophila, from yeast, from different mammals, including humans and mice, showing that in general we see a coupling of changes in gene expression and coding sequence, both at the specific gene level and also at the functional level. And in this particular system, it doesn't seem like that's happening, potentially because it's very young, and potentially about 1,000, 2,000, maybe as old as 10,000 years old. Okay, so I'd like to thank you all for your attention, and I think I have tons of time for questions. Yeah?
What's LD like in sunflower? Which I asked because do you think you could get SNPs within genes that are in the just
Yeah, so that's a good question. In general, so Helianthus annuus has a huge effective population size. We see LD decaying quite rapidly, and it's an obligate outcrosser. But this particular species has relatively small population and is relatively new, so I would expect to see hitchhiking occurring here. Yeah. So this particular approach, you wouldn't necessarily be able to identify the specific genes, and they would have to be expressed in seedlings. And, yeah.
So I actually did do the last question, which might be the same thing, but have you thought about looking later in the life cycle? Because when you sampled seedlings, maybe the things that are really important are actually expressed.
Yes, for sure. So this was definitely a sort of fun first pass side project, but this system I would love to develop further and look further at both phenotypic changes and how those relate to genotypic changes, gene expression changes, and also how those relate to the actually adaptation and fitness in the wild. Because the environments in which these 2 plants are quite different, grow are quite different. Okay, thank you guys. And we have about 2 minutes till the next talk, but please stick around.